Hello everybody, welcome to week 2 tutorial session. For the week 2 we are having two parts. One is the two different problems more specifically. The first problem we are addressing using a color coding method, and the second part of the second problem we are addressing using the logistic regression method. We will be discussing two different problems in each of these methods. First of all, for the color coding method, we'll discuss a problem and then we'll go to living. We will be discussing the problem on logistic regression. Okay. So I'll share the screen and we'll start discussing the problem for the color coding. Okay, so here's the problem for the here's the color coding problem. So we had the problem is something like this. This is an algorithmic problem. This is the problem actually. This problem is actually there in the paper color coding paper that we have shared with you. The paper which introduced how we can integrate color coding in computational biology. They have discussed the exact same problem that has been discussed in the video lectures. This problem is another a variant of the same problem that is being discussed in the lectures. This is also there in the paper. So you can look into the paper to understand the problem in further details, or for any more details, you can look into the paper. So what this paper states is, we have a I'll just give a brief summary of what we had and what is the new variant in the problem. Okay, so in the original problem, what we had is we had a uh, s- set of sources and set of target genes, right? We had a set of receptors that we called as S, and then we had a set of target genes G1, G2 that we called it as we used to call it as T. So we want to find all k length paths. We want to find k length path from any source, any node in S to a node V or G in T. Okay, to a node G in T, we want to find the k length paths, all k length paths from any node in S to this node G. Okay, and as we know, GRNs are directed, so obviously we want the edges to be in this direction from S to T, right? And now the thing is, we wanted to compute the maximum weight path, the path with the maximum weight. And because we had defined the weight using the probability, that is the probability of this particular edge is something, the probability of information transmission of this edge is something else, and so on. The max, we wanted the maximum weight because the weight of, we had defined the weight of a path as the sum of these probability values, and that's why we wanted the maximum weight of the path. Okay, so we had an optimal substructure, maximum weighted path. So we had the optimal this was the optimal substructure. We had a two-dimensional matrix where these V's, this was V, sorry, okay. So these V's formed the rows, and every subset of the different, every possible subset of k length of k colors, at most k colors. We used different colors to actually, the algorithm was something like we used colors to color every node in the network, and then every possible subset of the colors at most of at size at most k, okay, they form the columns in this GP matrix. And we computed the weight of this, computed the number of paths, or computed the maximum weight of a path to this V using this set of colors and stored it in the DP matrix. Okay, so that was the color coding algorithm that we had, and this was the corresponding optimal substructure. So now this variant of the problem, what it says is, apart from these two, let's say this is S and this is the target set of genes, I'm going to call it as G temporarily. Okay, for this particular problem, we are going to take it as capital G. Okay, the target set of genes. Apart from this two, we are having another set called as T. Okay, this contains a bunch of proteins. Okay, T1 to Tm. Okay, and what we are asked is we want to find a path with maximum weight, but the condition is there should be at least X proteins and at most Y proteins from T. Okay, so uh, what the information that is given to us is to do this. What we want to do is we want to modify, we have to write an optimal substructure for this. In other words, we have to design an algorithm, but for that, because we already know that this will be a DB algorithm, we can just do with writing the optimal substructure, and then we can convert the optimal substructure to a algorithm as and when required. Okay, so all we need to do is write the optimal substructure. So for the optimal substructure, what we are going to do is we are going to use a three-dimensional DP matrix instead of a two-dimensional DP matrix here. So apart from the information V and X, right, which is exactly the same as the previously what we had, apart from V and X, we are going to use another dimension which stores the which store like another dimension which stores the number of proteins, which doesn't exact not exactly the number of proteins, but it is a count on how many proteins we are including from T in that path. Okay, so here we will be having WVX comma P. Here V and X are basically the same as the maximum weight of, it stores the maximum weight of a path. This particular cell VXP stores the maximum weight of a path. So that path must be having length of mod X. It should end at P, and it contains a vertex of each color in X, and along with that there are exactly P proteins from T. Okay, so that is what we have to we are having. So given this information, what is the correct optimal substructure? So what we are doing is, for the solution, coming to the solution, what we happen is we have this W equals to VXP. So we want to compute this value. What we want to compute is, we want to compute what this value is if I am having P minus, if I am including some protein. Let's say V is a protein itself. V is in T. If V is in T, then what should be the optimal substructure? And if V is not in T, then what is the optimal substructure? Here we are going through via every node, right? So that is why we have to take these two cases into. So for the optimal substructure, we have to look into these two cases, whether V is in T or V is not in T. So for that scenario, we will be doing this. So this is actually the optimal substructure. This is the optimal substructure. What happens is we look into, if we not in T, then what happens, and if V is in T, then what will happen. So I'm going to pause it a little bit, and before going into details. So what happens is, if V is in T, what happens? My P will now include V also. I am including exactly P proteins. So when I am considering, I am taking exactly P proteins, and from on that V is also one of those proteins. So I have to look in the lower dimension. Okay. So what I will be looking into is will be in the lower dimension. So I have to look in the P minus 1 dimension. Okay. So now what will I look for in the P minus 1 dimension? I am using this X, sorry, using this V. So I have to look for the U. So I have to find the maximum value of U in the lower dimension. It's exactly the same as in the previous, in the original color coding algorithm that we had discussed. So we want to find the weight of Ux minus C of V, the way we used to compute it. So it will be exactly the same, but here the difference will be, because our V is, if our V is in T, then we will be looking into on lower dimension. And if V is not in T, then we will be looking into the same dimension. Because V is not a protein, so we will not be including V in P. So which means that we are already having P proteins, so we'll go and look in the same dimension itself. Okay, so this is the only difference between the original color coding algorithm that we have seen in the lectures and this color coding algorithm. It basically depends on these are the two extra constraints that we have to check whether V is in T or not. So apart from that, there isn't much difference between this particular problem and the other problem. Okay, so this was the first question. So the second question will be on logistic regression, and Levin will continue on that. Stop share. Levin, you want to continue? Hello?
Just give me a minute. So this do not complete yet. So I'll go through so that we are to maybe in the next session I will do the thing of that and the actual logistic regression part. So I start with this. So let me share here. I'll give a brief of the problem that we are trying to tackle. I hope my screen is visible. Yeah. So this is the paper that kind of introduces this protein interaction. I mean, this protein interaction with this logistic regression. So you can find this supplementary documents which specify skills about the logistic regression methods that they are using, and actually this is that one. So this is estimation of interaction probability using logistic regression. So this is what trying to build. So apparently the data set that they were using the regular paper, so the database are not currently available in the format that they use. I'm trying to obtain it, so I'll do that part also. Before we'll go through what are the steps that we are trying to do to achieve this this track problem, right? So they're modeling this using a logistic regression model, and for a given species, we are taking a species and we are representing this probability of interaction using this three features that we are observing. So one of the features is the number of times the interaction between the proteins observed, and the second one is the Pearson correlation coefficient, and third one is the small world Pearson coefficient. So these three things are what we are trying. These are the features that we are trying to extract in interaction data, right? So this is the basic formula for the Pearson correlation coefficient. And we once we have that, we will be able to find. So once we have the x and y's as pairs, we'll be able to find the number of times the interaction happens. Basically, we are going to count how much time this protein observed or the pair was observed. Particular pair or observed. And again, it's a Pearson correlation coefficient between the expressions of these. So we need this of this genes, this expression measurements for the corresponding unit. So we have given a particular pair of genes, we have to find the coefficient in this expression gene experiments genes, right? So we need the expression measurements to compute this for a particular pair of x and y. And this Pearson computing Pearson coefficient can be easily done with once you have this vectors, can be easily do this using uh, libraries. Then the third one is small world clustering coefficient that we need to compute. So to do this, so we need to construct this network of interactions. So basically, construct a graph. So in Python, easier way to do this section of this network is use this library. So for each pair, like the paragraph, we add this work and add an edge between each of these pairs. After that, we denote the set of proteins that interact with them by using NV and NW. So these are the pair. V and W are the pair of these. Then the small world clustering coefficient is given by formula. So this formula is quite complicated as it involves some binomial operations and so. But once it's mathematically implementable. Okay. So and then we find this clustering coefficient UV, UW, and this coefficient, and the number of action happens. So all for a particular pair of protein. So for each pair of protein, we'll have three features. For each pair of protein, we'll have three features: x1, x2, x3. Actually, this times and this one you can call this coefficient x and y. Similar to that, what are the variables that you consider? Here, the interaction probability that particular pair with the two interactions given by largest model. So these three features will be the input to the model, and using the scikit-learn, once we prepare this set of x, we can and y, these two interaction probabilities that we measure. So this we get from the data set, from the already existing data set. We'll get the measure in probabilities from. So we compute this from the data sets and also this available, which is the measured value, and using that compute this. We want that using this logistic model. This is the logistic regression method. Behind this using the training data. Right. So they were used is like very old, which is not to the coding part of this. Put the data and show you how this whole operation can be done. So let me go through this data sets first. So this data called Byte, which provides you with this track data. Oops. So here you can find the this tab separated format like here by containing this interaction and genes from different organs, right? I kind of loaded it into this thing, and so this is how data looks like. So you can find the gene ID, the other ID that they use, about the genes, and then there will be this. So you have these features, this interaction data, and there is a score. Yeah, so the score gives us the interaction thing around here. So let me just take these three fields, which is the gene ID, and still score. So this is basically I filtered out only the things involved in humans and in the organisms, right? So, most of the course is not clear. Let's say so now we get a filter with non-empty scores, right? So the true interaction score between these two genes can be found in this table out from the whole data. This is what we basically need. And now with this we can find the count of occurrence of this error in different cases, right? So that can be done using by operation like this. You can basically go by this interaction data and then count it. So you get the interaction thing in the data set. And then to compute the Pearson coefficient, we need the gene expression for this particular genes. So how do we find it? So actually the data set that's kind of not in a very easy format to do. So that's the part where we can't. So this they provide the data principle in phase like this. So the GID, and then you can find the gene expression of a particular gene, which is this one. Let's also take this this one, official simple interact these two cards. This will help us confirm whether they are find a particular gene or not. Like the names of yeah, let's do this. Yeah. So let's take this ID. So this is set this NPJ GEO, which is genomic expression which has all the genomes around there. Okay, so you give video and give search. We'll get the name of the particular gene and the details about it, which is kind of IPC this thing, right? And here we can find the gene expression. So the gene expression, like it's of a vector. You see the different columns around here. This is the vector produced for each of this column. So similarly, we need to have this particular expression for this corresponding gene. Let's find this one. So, like this particular thing might need scraping that to get all this. So basically, this can be downloaded as this gene thing can be downloaded as a expression. So this can be downloaded as a text file. Let's see. We need to pass gene expression thing, and then we have to store it in a like high-explore format. So for this gene, you can find the gene expression over here. The same kind of effect. So this is basically the reflector. What we do is find the Pearson relation between these two gene connections. So we need to do a form description to get all gene connections that haven't done yet. So once we get all these gene IDs, this complete and with the remaining of this. So once we get this part, computing Pearson correlation is pretty straightforward. We can use this from stats to compute this Pearson coefficient. Then for the small building coefficient, this network X library can be used. So here we can create a graph, the pairs, and then compute the thing over it. So this is a ChatGPT generated example. So we get this gene expression, like once we scrape this gene expression data, we'll be able to use this 3 fields and extract this each. And then the logistic regression part is like pretty easy. If I pay dot logistic regression, you will be able. So maybe once in the next session, once I extract this features, I will be able to show this with the plots. Okay. Till now.
Yeah, license and everything, right? So at the moment, I'm not going to do that. I want to go. So it's
Yeah. Hello.
Hello.
Yeah. So
Hello? Sorry, the voice is little weird. I couldn't hear properly in the end.
okay. Okay.
you able to hear me the whole time, or
No, only the last portion of it, before the logistic
was
regression
it
portion.
the code?
Yeah, the code. Can you hear me?
Yeah, I can hear you. Okay, let me show the final part. So, did you understand where we are getting the data from, and what's the data that we have?
Yeah, to some extent. Anyhow, I'll be watching the recording also again.
Yeah. Basic idea is this data set called BioGrid. There is this data called the BioGrid provides us with the protein interaction data.
Okay.
Downloading this tab separated CSV file here. And that's the main DF that I have loaded in here. So this has information about the protein interaction, the protein gene and the interaction things. I have like a filtered subset of with only the necessary columns that we need, the gene ID, and then the source, interaction source.
Yeah.
So with this, we also need the gene expression, right? So this data set doesn't have the whole gene expression. So to get the gene expression, there is another website called Gene Expression Omnibus. This genome expression omnibus.
That's what was suggesting because
Okay.
So here basically I can search this with ID, you can get the gene expression manually.
ID, then you find it.
You might have to, like this is the gene ID, right? With this gene, you'll be able to get the gene expression from this website. So we need to create this expression. So it's basically downloadable. We have to do all expressions because to find the piece of calculation, we need this one.
Okay. It means like the G at the 10-week, this much RPKM is this much of gene is expressed or something like that, or that particular.
Yeah, so this is the gene ID, right? So this is the ID of the particular gene, and this is kind of the name of that genes.
Okay.
And you can search this one with the gene ID, and it will give you all the details about that particular gene. This is the gene, and you will get all the details about the particular gene in various measurements.
Okay, okay. How much of the gene is expressed in various parts? Okay.
So how many samples they took and that
Okay.
thing, right? So from this gene expression of some different numbers that you have, different measurements, right?
Yeah.
So this gene expression, comparing with the other gene to find a correlation coefficient, how it's correlated.
Okay, okay. I get that. I saw
Is-
that. Yeah.
After that, this is the correlation part. We need this gene expression, and for the other part, like the interaction thing, we need to build a network X graph with this corresponding pairs as edges, and with that we'll be able to find the small world clustering coefficient.
Yeah, I was following all this. Yes.
So this one is like the pairs, right? So we use that as the edges stuff. Okay.
Yes.
So we build
yes.
that graph and then we use this clustering coefficients from this library, this network X library.
Yes.
So then we basically will get a thing like these three features that we saw over here. We'll get these
Yeah.
three parts for a particular pair of proteins. So we'll get these three features. And the y value will be this score that we have. So from this data set, this is the y value that we are going to predict.
Yeah.
Okay.
Yeah.
Okay. So once that gene expression, I'll be able to training and do this in the next session.
Okay. 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 Yeah, I get
Oh.
that. Yeah, thank you. Thank you very much.
And then the next
I don't have any questions now.
Okay, okay. If this is done, then I'll end the session. Okay, I'll stop streaming and end the session. Okay, thanks. Thanks
Yeah.
everybody for
Thank you.
joining. Thank you. Oh, Levin.
Yeah.